Hello students, I'm going to do a quick review before the lab practical number 2. As you can see, I'm not fully operational, but because this is a 50 question test, I want you guys to be prepared. So really quick, we'll go through the lab 8, 9, 10, 11, 12, and 13. There's a lot of questions that we can go over. So what happens to a plant cell in a hypertonic solution? So hypertonic solution, plant cells plasmolyze. Fusion is a movement of solutes from an area of high concentration to low concentration, right? So you want to take a high concentration down to a low concentration. Is diffusion an example of passive or active transport? Diffusion is passive transport, right? Because it doesn't need energy in order to let that transport happen. Under what conditions is a plant cell turgid? That would be hypotonic solution. For red blood cells, which solution is hypotonic? We have 3 different options. We have distilled water, we have 10% NaCl, and we have 0.9% NaCl. So if it's hypotonic, it's going to be in distilled water. And then if it's hypertonic, it's going to be in 10% NaCl. If a plant cell is turgid, will it be standing upright or wilted? Well, if it's turgid, it's going to be upright, right? That means that it's in a hypotonic solution. It's given water. But if it's plasmolyzed, it's going to be wilted. Because the walls have, or the plasma cell membrane has enclosed on itself. The walls have no pressure to hold it from falling apart. So like if you were to sit on a cardboard box, if it's empty, you can just sit on it and it's gonna flatten. But if it's full of, let's say, like bricks, if you sit on it, it's not going to flatten. Make sure that you know what the differences are between the tubes of red blood cells. Okay? How they look. Right? So if one appears to be a cloudy red, one appears to be a clear red. Which one is crenated, which one is hemolyzed. All right. So test tube A appears to be cloudy red. Test tube B appears to be clear red. Which one of those is crenated? So the crenated one will appear cloudy red. So if test tube A is cloudy red, then it's test tube A that is crenated. Make sure you know the difference between solvents and solutes. Okay? The solute goes into the solvent, right? So whatever you're pouring into it is the solute, and whatever is there is the solvent. All right, let's move on to lab number 9. This one is the one that you should have done your lab on, so you should know a lot about this, your lab report. So you should know about this more than the other ones. So how does an enzyme affect a chemical reaction? It should lower the activation energy. So it lowers the activation energy, allowing for chemical reactions to occur more readily. You should know why enzymes are reusable. They're not part of the chemical reaction. They just facilitate that activation energy. They bring the molecules closer together so that they can facilitate that activation energy. What type of macromolecule are most enzymes? Most of them are polypeptides. Right, they are mostly made up of multiple amino acid chains brought together. They are polypeptides, they are proteins. What is the name of the enzyme you used in your experiments? So in the experiment on lab number 9, what is the name of the enzyme? Should be catalase. Right? Whenever you mashed up the potato, you brought out the catalase within the potato. You should know what's able to denature an enzyme. So pH, temperature, also chemical conditions. Okay. What substrate does catalase enzyme bind to? Catalase should bind to peroxide. Not specifically hydrogen peroxide, but that's what we used. So whenever you have a peroxide in there, catalase will take off an oxygen. So how do you know reaction is taking place? When oxygen is released from the reaction. What do we see at 100 degrees Celsius? What happened to that enzyme? So the enzyme failed to produce oxygen. Or you can say it was denatured. What happened to the enzyme hydrochloric acid? Technically it's supposed to have failed, but what happens is it tends to run very quickly and then it drops off very quickly because the enzyme will shoot up a very high amount of oxygen and then it'll drop off once it becomes denatured. So it does become denatured. All right, that's all of lab number 9. Let's move on to lab 10. So byproducts, whenever we talk about a byproduct, it's not part of that process anymore. So a byproduct of us, of mitochondria, is carbon dioxide. It's no longer use of us anymore because we don't use carbon dioxide anymore, we breathe it out. So a byproduct of photosynthesis would be oxygen and sugar. Where do the reactions of the Calvin cycle take place? So in the chloroplast, where does this take place? So this is going to be in the stroma. Right? So it's going to be right outside the thylakoid membranes. So then, if the Calvin cycle occurs outside the thylakoid, the light reactions will occur in the thylakoid membranes. Why is it beneficial for plants to have different pigments? So, plants have different pigments for a variety of reasons, but in the sense of photosynthesis, plants can pick up different wavelengths of light. What gas is necessary for photosynthesis to occur? Carbon dioxide, right? Because otherwise it wouldn't be able to take in that carbon and fix it into sugars. What is the laboratory technique that we use to separate those different pigments from each other? So we use those little strips, or I showed you those little strips that we used in the lab. It's chromatography. So in chromatography, which pigment was the most polar out of all of those pigments? It was one of the chlorophylls. I believe it was chlorophyll B. And then which one was the most non-polar? Right? The most non-polar being the top one, and that was carotene. What does sodium bicarbonate provide for photosynthesis to occur? Sodium bicarbonate provides carbonation, it provides CO2. In the spinach leaf experiment, what causes the discs to rise? So that would be oxygen trapped within the discs. Oxygen trapped within the leaf discs. Right, so when that CO2 is brought into the leaf discs, photosynthesis takes hold of that CO2 and converts it into oxygen. Right, and so it can traverse that into oxygen and then it'll float up. Okay, let's move on to lab 11. We talk about fermentation. First of all, is fermentation more or less efficient than cellular respiration? It is less efficient. So why would a cell use fermentation rather than cellular respiration? So cellular respiration is only occurring when there's oxygen around, right? So fermentation is a way to get around that to survive if there's no oxygen around. What organism was used in the fermentation experiment? So this experiment used yeast, Saccharomyces cerevisium, I think. Anyway, it's a type of Saccharomyces. What gas is produced by fermentation? Carbon dioxide. Just like every other cellular respiration, carbon dioxide is produced by fermentation. In the fermentation experiment, which of the three sugars was a disaccharide? And I went through them in the experiment to tell you this one's a monosaccharide, this one's a disaccharide, this one's a polysaccharide. It's fructose, by the way. Okay, so let's move on to lab 12. What phase of cell division does DNA replication occur? That's interphase, S phase. How many pairs of chromosomes would a human daughter cell have at the end of mitosis? So a human daughter cell would have 2 pairs of chromosomes at the end of mitosis, right? It would be diploid. What is the process called when cytoplasm divides? That's cytokinesis. It literally means cell moving. What phase of cell division do sister chromatids become daughter chromosomes? That's part of anaphase, right? Because they've split apart now. So they're sister chromatids here, but when they split apart, they're daughter chromosomes. You should know what a cleavage furrow is and what process a cleavage furrow is involved in. How many rounds of division does it take to produce the daughter cells of mitosis? It only takes 1 round of division in mitosis. Okay? 1 division. And it goes through 1 division. Okay, let's get into lab 13. What is a somatic cell? This is something that many of you didn't quite understand. A somatic cell is every other cell except the sex cells. Okay? These are your body cells. How many chromosomes are present in the eggs and sperm of a human? So how many chromosomes are present? That's 23. Right? Usually there's 46 in each of our cells. There's only 23 in the egg and sperm cells. Are the gametes haploid or diploid? They're haploid, right? Because there's only half the number of chromosomes in the sperm and egg than there are in the rest of the somatic cells. What phase of meiosis does crossing over take place? That's in prophase 1. In a sperm cell, where is the DNA located? This would be in the head of the sperm cell. So the sperm has a head and a tail. It's only in the head. How many rounds of division does it take to produce daughter cells of meiosis? So this is 2 rounds of division. Mitosis had 1 round of division. Meiosis has 2 rounds of division. You should know how to define oogenesis and spermatogenesis. Right, they're both what they sound like. Oogenesis is the development of egg cells. Spermatogenesis is the development of sperm cells. That's about it for the review. I hope that this at least gave you some insight to what the test is going to be like. I did go over quite a bit of the questionnaire, so please just go over that and go over some of the notes and everything. Okay? So, hope you do well on the test, and I will see you later.